Hello everyone, my name is Colin Ballard. I am a first-year PhD student at Case Western Reserve University, currently working in Dr. Thomas Gerken's lab. In our lab, we study the initiation and modulation of Mucin-Type O-Glycosylation, and today I'll be discussing to you about our discovery that substrate charge distribution modulates the activity of polypeptide GalNAc transferases responsible for initiating Mucin-Type O-Glycosylation. Mucin-Type O-Glycosylation is the transfer of an alpha GalNAc from UDP GalNAc onto the hydroxyl groups of serine and threonine residues within a peptide substrate via N-acetylgalactosamine transferases. We have seen that there are many unique specificities for these transferases for serine and threonine residues, peptide sequence, prior, remote, and or neighboring glycosylation. Some of our recent studies of FGF23 and the splice variants of 5-pCant9, we saw that flanking charge residues altered these rates of glycosylation. So in order to systematically address the rules of flanking charge, we obtained a series of peptide substrates possessing all the possible N and C terminal combinations of flanking positive, neutral, and negative charge. We also created homology models for the GalNAc transferases that did not have a crystal structure available, utilizing three model templates seen in Figure 3. These model templates were based on the orientation of the lectin domain to the catalytic domain. GalNAc T3 seen here is the 6s24 structure, in which we like to call it the left compact structure. The GalNAc T2's 2FFU, the extended structure, and the GalNAc T2 5AJP structure, or we call the right compact structure. All the peptides on these structures were also overlaid onto our homology models and our electrostatic models. Electrostatic analysis was also conducted using the APBS extension in PyMol, and in Figure 4 we see the effects of flanking charge residues on GalNAc activity. In Figure A, these are the GalNAc transferases with negatively charged substrate preferences, in which column 1 we have all of our peptides from positive to negative. In column 2, it's the same data seen in column 1, in which the charge in either the N and C terminal portion of the peptide remains constant while the other one changes. And in column 3, we see the electrostatic map of our GalNAc transferase with the PDB of the structure or the template used to obtain that model. B shows the transferases with positively charged substrate preferences, and C is the transferases with unique substrate charge preferences, in which they demonstrate no definitive pattern or preference for peptides, and the electrostatic charge map leads us to no definitive conclusions. In Figure 5, we tested two of our enzymes, the TG GalNAc T3 and the H GalNAc T2, and we saw that these Vmax and Km values drastically changed based on the charged substrate preferences of these two enzymes. T3 is represented in Figure 5a, and T2 is represented in Figure 5b. These results further enhance our conclusions drawn from Figure 4. In conclusion, our experimental data has led to the discovery of another factor in initiation of Mucin-Type O-Glycosylation. These results are further supported by our electrostatic modeling, which correlates well with our obtained data, and our kinetic analysis. With the discovery, GalNAc T isoform specificity and its role in disease will be better understood. Future experiments are underway to see how flanking charge residues affect the continued Mucin-Type O-Glycosylation of adding additional sugars to GalNAc residues with the analysis of these transferases, such as Core1, the Core3, and the ST6 GalNAc 1 and 2 transferases. I want to thank everybody for listening to my talk and thank our collaborators for making our research possible. Thank you and have a great day.